Today we're going to start Heredity 2, and we're going to take a look at an overview of what we're going to be learning in this part of the unit. We're going to focus on how characteristics of life are passed down from one generation to the next. So we're going to take that information from Heredity 1, where we were really trying to understand how a parent can pass on a copy of their genes to their offspring. And we're going to just take a look in detail what those instructions look like. So we're going to start off with DNA structure and then focus on how DNA can make a copy of itself. And then it's going to switch gears where we're going to get into what's known as the central dogma of molecular biology, or protein synthesis. So how do we take DNA and make proteins from that DNA? And then take a look at mutation, what happens if something goes wrong in either DNA replication or in protein synthesis. And then also take a look at, where we are right now with biotechnology or genetic engineering. So using this knowledge and applying it to science. If we take a look at this picture, I love pictures that try to show how these words actually go together. And if you take a look here, we have a homologous pair of, actually you have sister chromatids. So they're attached here at the centromere. And it's actually showing that this chromosome is made up of DNA, and different segments of DNA are called genes. And if you look at a gene even closer, you're going to focus on the nitrogen bases to see what they represent. On the next page in our packet, depending upon what course you're taking, you have some information about scientists either filled out for you or you had an assignment that you researched some of these scientists. But either way, if you want to take a look, there's a timeline that you can check, but you do not have to memorize the dates. The main scientists that you're responsible for are Erwin Chargaff in the 1950s, and then Watson, Crick, and Franklin in 1953. But what I always like to point out to students is if you look how many scientists actually contributed to what we know today about DNA, about the structure of DNA. And this doesn't stop in 1953. Again, if you check out this timeline, you'll see how many scientists continue to contribute the pieces of the puzzle to understand what makes us, what makes us human, how we're related to other organisms. But if we look at Chargaff, he was a chemist who basically discovered the nitrogen bases, and he kind of is credited with the base pair rules that we're going to look at in the next slide, which is A equals T and G equals C. Watson and Crick actually pulled a lot of this together and were credited with the discovery of DNA structure being a double helix. They used information from Rosalind Franklin, who used X-rays to learn about the structure of DNA. So she actually provided the work, the data. So some things that you should know about DNA: it stands for deoxyribonucleic acid. So as a class, we kind of came up with some facts or some important things to remember about DNA. So if you want to take a chance to write those down and pause the video and write those down. We also took a look at this image. And again, I love these images that kind of show how all these terms are related. So, inside your arm, there's a cell. Inside that cell, there's a nucleus. That's where our chromosomes are located, and our chromosomes are made up of DNA. And the same cell right here in your arm, and the same cell in your brain, and the same cell in your foot, they all contain the same exact DNA. But what we talk about is that some of those genes, some of that DNA is being used in the arm and it's not being used in the brain, and vice versa. Sometimes the DNA is being used in the brain and not in the arm. So when we get to, figuring out how proteins are made, they'll make a little more sense, but it is important for you to know that the same DNA is in every cell in your body. All right, so flashback to Unit 2 when we talked about organic compounds. One of the organic compounds was a nucleic acid. And there were three examples. DNA and RNA are two nucleic acids that we're going to talk about in this unit. The monomer of a nucleic acid is called a nucleotide. So the nucleotide of DNA, or the building block of DNA, is a nucleotide that consists of phosphate, deoxyribose, and a base. The deoxyribose, if you look at the ose ending, you will see that it's usually a hint that it's sugar. And the bases that are involved in DNA are adenine, thymine, guanine, and cytosine. Another thing to point out about the structure of DNA is that these nucleotides line up what is called anti-parallel. Okay, they are not quite parallel to each other or even to each other. And if you take a look here at this end is the sugar, whereas on this side, the end is the phosphate. So they're kind of running opposite of each other. Aaron Chargaff was the Austrian-American chemist who discovered that the amount of adenine always equals the amount of thymine, the amount of guanine always equals the amount of cytosine. So, and this could go, thymine is going to equal adenine, cytosine equals guanine. It doesn't matter which way you write this. These are, adenine and guanine are two purines, and thymine and cytosine are pyrimidines. So originally scientists maybe thought that, the G's and the A's paired up, but I always remember at, so that it helps me remember they go together. So really, when you know the composition of one of these, you can figure out the others mathematically. So if we start here with 20 adenine, we know that thymine has to be equal. If you take 20 plus 20, which is 40, subtract that from 100, you get 60. And 60 has to be evenly split between guanine and cytosine. So this base pair rule is what helps you. Adenine, thymine, sorry, my pen isn't working real well here, helps you actually match up to make the other side of DNA. So if you can take a minute and pause and try to match the other side up. You pause the video, I'm going to reveal the answers here. So if you are going to take a second to do that, pause it now and write those in. And here is the answers. So A goes with T goes with A goes with T goes with A, C, G, A, T, A, G, C, C, G, C, C, C. So you should be comfortable with making sure that you have followed the base pair rule to make the other side of DNA. This is also going to come in handy when we do DNA replication. But before we get there, let's just make sure we understand that the combination or the order of the A, T's, G's, and C's are going to determine what traits you have. So everyone's going to have a gene for hair color, but the allele or what combination they have is going to be different. It's going to actually determine what color their hair is. So this goes back to the last unit where we were learning about genetics, and we talked about, what alleles, what versions or what alleles you have, and the alleles are these actual letters. But we didn't talk about that because we weren't there yet. But this is what that represents. Okay, and finally, DNA replication. So in order for every cell to have an exact copy of DNA, it needs to replicate itself before it divides. So going back to Unit 4, the DNA replicates during the S phase of the cell cycle. DNA synthesis is another name for DNA replication. It's called semi-conservative because half of the original strand gets into a new strand, and so does the other half is another new strand of DNA. The important enzymes to know are helicase, which is an enzyme right here that's going to unzip the DNA. And we call that area the replication fork. And you'll see this is happening continuously along the DNA during the S phase. And then there's another enzyme called DNA polymerase, both sides also. This enzyme is going to add nucleotides to the growing new strand of DNA. When it's all said and done, you'll have 2 exact copies of DNA to go into the new cells. So take a look at this last page. I'm going to reveal the answers. You can pause the video and check your answers. And I hope that helps you understand DNA structure and DNA replication.